tell you genetic diseases in these 3 sessions. Thanks for your patience in advance. Let's talk about the human genome. Human genome discovery was a turning point in the medicine and the biology. As you know, human genome length is approximately 3.3 billion nucleotides. Just only 1.5% of the genome is translated to the protein. The other parts is non-coding sequences. You can describe the genome as a library, a chromosome as a book, and also a gene as a picture of the book. Proteins provide the building blocks and machinery required for assembling cells, tissues, and the organism. It's the non-coding region of the genome that acts as the architecture and planning. So the non-coding, the rest of 98.5% of the genome has a regulatory role, which is very important in the human genome. The proteins variously function as enzymes, structural components, and signaling molecules. This is the structure of the gene, 5' UTR, intron, exon, and also 3' UTR. Non-coding part of the DNA approximately constitutes 98.5% of the human genome and does not code the protein. The difference between the human being and other creatures is in this part of the genome. There are different types of non-protein coding DNA sequencing, including promoter and enhancers that bind transcription factors, binding sites for protein that organize and maintain higher order of chromatin structures, non-coding regulatory RNA such as microRNA and long non-coding RNA, jumping genes or transposons which can move around various sites in the genome and are implicated in gene regulation and chromatin modifications, special structure regions of the gene that play a role in the stability of the chromosomes like telomere and centromeres. Many genetic variations, polymorphism, are associated with diseases located in the non-coding, non-protein coding regions of the genome. Promoters is a region of DNA that initiates transcription of the gene. It can be about 100 to 1,000 base pairs. Enhancers is a short, approximately 50 to 1,500 base pair region of the DNA, increase the likelihood that transcription of a particular gene. Jumping genes or transposons can move around various sites in the genome and are implicated in the gene regulation and chromatin organization. So it is a very sophisticated process, regulation of the gene expression. MicroRNA and long non-coding RNA. MicroRNA, as its name show, are relatively short RNA, approximately 22 nucleotides, that function primarily to modulate the transcription of target proteins into the corresponding protein. It causes post-translational suppression or activation of the gene expression, especially suppression and degradation. Long non-coding RNA are approximately 200 nucleotides in length and modulate gene expression. They play a role, for example, in X chromosome in activation. Most common form of DNA variants in the human genome are single nucleotide polymorphism. Nowadays, they are called variants, not polymorphism. Polymorphism is a population genetic definition. For example, if in a genetic pool in a region, frequency of a mutation is more than 1%, it is considered polymorphism. It is not something very rare. But nowadays, every change in a single nucleotide is called a variant. it can be polymorphism, non-polymorphism. So it's better to call it single nucleotide variant or copy number variations, which include larger changes in the DNA sequences. A SNP or single nucleotide polymorphism are variants at a single nucleotide position in DNA. For example, A changes to G. It is called a SNP or SNP, single nucleotide variant. The SNPs can be neutral variants with no effect on the, for example, codon changes to codon of the same amino acid, no change in the protein sequence, so it has no change in gene function or effect. The SNPs located in non-coding regions can occur in regulatory elements in the genome, thereby altering gene expression in
خدمت شما باشه این در زمان که قیام بود این حضوریات رو اگر انجام بدید ممنونه من این بغل آموزش هستم
thereby altering gene expression in instances that the SNP can have a direct influence on disease susceptibility. Each human being, we have over 6 million SNPs, and many of wide variation in the between the persons can be due to these SNPs. And now let's talk about the copy number variation. Copy number variations are larger changes of approximately more than 50 base pairs to thousands base pairs in the gene sequence. The number of copies of a specific segment of DNA varies among different individuals. It can be deletion, duplication, inversion, or insertion of a segment of the gene. They may or may not contain a gene. Approximately 50% of CNV or copy number variants involve gene coding sequences. CNV may underlie a large portion of human phenotype diversity. So many of the human phenotype diversity is due to the copy number variations. These structural differences may have come about through duplications, deletions, or other changes, and can affect long stretches of the DNA. Alterations in DNA sequence cannot by themselves explain diversity of phenotypes in the human population, or even the difference between two identical twins. They have the same DNA sequence, but they are morphologically different from each other. You can, for example, make a difference between these two identical twins by shape. So the difference are not just because of DNA variants and variations in the DNA sequence. How you can change phenotype without changing the genome? The answer relies in the epigenetic. Okay, another definition: epigenetic literally means above the gene, is defined as a heritable change in the gene expression without the change in the DNA sequence. So it is called epigenetic. It is controlled by a collection of protein and molecules that manipulate DNA to turn genes on or off. Epigenetic changes affect gene expression to turn genes on or off. Unlike the genetic changes or mutation, epigenetic changes are responsible and do not change the sequence, reversible and do not change sequence of DNA bases, but they can change how your body reads the DNA sequence. There are different mechanisms for the epigenetics. First of them is DNA methylation. The DNA methylation turn off a gene and suppress the gene expression. Vice versa, DNA acetylation activates the gene expression. So a protein modulation affects its expression. Different mechanism of epigenetics. The level of DNA methylation decreases with for example, newborn does not. Question? The level of DNA methylation decreases with gene. For example, a newborn has the highest level of methylated DNA, but a very old man do not have such methylated DNA. So let's return to the basic definitions in the genetics. Have you passed the genetic course? Not yet. Commonly used terms: hereditary, familial, congenital, somatic cells and gametes, chromosomes, chromatids, and centromere in the types. Okay. Hereditary means derived from one of the parents and are transmitted in the gametes through the generation to the offspring, so it is familial. So hereditary is the same as familial in the genetic. The congenital influence present at the birth. Okay? Some congenital diseases are not genetic, and not all genetic disorders are congenital. The examples are, for example, the hereditary syphilis. The congenital syphilis is not genetic, it is an infectious disorder. And also the autosomal dominant disorders like Huntington's disease will happen in the adulthood, so they are not all congenital. Somatic cells versus gamete cells. Somatic cells are non-sex chromosomes, and each gamete contains only 23 chromosomes instead of the 46 chromosomes. The difference, between somatic and nothing very important. Somatic cells have 2 copies of chromosome, but the gametes only have 1 copy of the chromosome. In the mitotic metaphases, we can see 2 main structures in chromosome by light microscopy: a chromatid and a centromere. Pictures of chromatid, centromere, telomere. 2 chromatids making a chromosome are referred to as 2 sister chromosomes. 2 chromatids of homologous chromosome are non-sister chromatids. What are homologous chromosomes? 2 copies of each chromosome come from parents are identical. They are called homologous chromosomes. Chromosome structure, P stands for petit, means small. Short arm. The alphabet after P is Q, long arm, and also the centromere, telomere, the structure. Depending on the site of the centromere, the chromosomes are classified as metacentric, means the centromere is at the mid of the chromosome, submetacentric, not just in the mid, apocentric, and near the end, and telocentric, just at the end. The DNA structures composed of nucleotide. The nucleotide is composed of sugar base and phosphate. Chromosomes are rod-shaped bodies seen during metaphases. During metaphases, you have chromosomes in the most condensed form. Let's about the protein synthesis. Protein synthesis consists of 2 processes. Transcription takes place in the nucleus. The DNA is transcribed to the messenger RNA, and in the translation, the DNA is translated to the proteins. Transcription. RNA polymerase binds to a region of the gene called the promoter sequence. The strand is used as a template. It's called the template strand. And the strand of DNA is synthesized complementary to the template DNA. Processing of messenger RNA has 3 steps: splicing, 5' capping, and polyadenylation of 3'N. Splicing removes introns from the messenger RNA Introns are regions that do not code proteins. The remaining messenger RNA consists of only regions called exons. 5' capping and a methylated cap to the head of the messenger RNA, and it helps it stability and also binding of the ribosomes. Polyadenylation also added poly-A at the 3' end of the messenger RNA and helps it stability. In the translation, RNA is translated to the protein. Messenger RNA leaves the nucleus. It moves to a ribosome, which consists of rRNA and proteins. Translation has 3 steps: initiation, elongation, and termination. In the initiation, translational complex forms and tRNA brings the first amino acid in polypeptide chain to be bind to the starting codon of messenger RNA AUG. Elongation, tRNA bring amino acids one by one to add to polypeptide chain, and in the termination, release factor recognize the stop codon, translational complex dissociate, and completed polypeptide is released. Okay, let's talk about the nature of the genetic abnormalities. First, mutation refers to permanent changes in the DNA. Those that affect germs are transmitted to the progeny, to the offspring, and may give rise to an inherited disease. Mutation in somatic cells not transmitted to the offspring and progeny, and they can cause cancers or congenital malformations. Common examples of the gene mutations are point mutation, frame shift mutations, and trinucleotide repeat mutations. A mutation in enhancer or promoter areas can cause abnormal or no translation, for example, in hemolytic anemias. Mutations in intron can affect the messenger RNA expression. What is a point mutation? Point mutation is a substitution of a single nucleotide base by a different base, resulting in a replacement amino acid by another amino acid. It is called a missense mutation. If one amino acid is changed to another amino acid, it is a missense mutation. By contrast, certain point mutation can cause codon of an amino acid be changed to a stop codon. For example, it is called a nonsense mutation. The example for a substitution or missense mutation is the sickle cell anemia. You have read sickle cell anemia in the previous courses, I think. It's autosomal recessive disorders. Persons inherit 2 abnormal copies of the beta globin gene on chromosome 11. Hemoglobin A2 have 2 alpha chains and 2 beta chains. In sickle cell anemia, at least 1 beta globin is replaced by hemoglobin S. For sickle cell trait, you need 1 abnormal beta chain. For sickle cell disease, you need 2 abnormal beta chains. In these chains, CTC changed to CAC, and glutamate or glutamic acid is substituted by hydrophobic valine amino acid. It has various presentations. For example, anemia, pain crises, and also susceptibility to infections due to malfunction of the spleen. Mechanism of the sickle cell, glutamic acid to valine, it is a missense mutation. Let's talk about the frame shift mutation. Frame shift mutation occurs when the insertion and deletion of 1 or 2 base pair alters the reading frame. 1 or 2 bases, deletion or insertion. normal frame, 1 or 2 is added, the amino acid sequence is completely different. So with these, a frame shift mutation, most of frame shift mutations are pathogenic, they are disease causing. Let's talk about trinucleotide repeat mutations. Amplification of the sequence of trinucleotides is a type of mutation. These sequences mostly include guanine and cytosine. For example, Fragile X syndrome, prototypical of a category of this is disorder. In this disorder, CGG sequence, trinucleotide, is repeated more than 200 times in FMR gene. In normal population, this repeat in FMR gene is approximately 29 sequences. Another distinguished feature of trinucleotide repeat are dynamic changes. In each generation, especially in oogenesis, this sequence is amplified, is increased. As the trinucleotide repeat expansion grows, the growing triplet tract alters the gene expression and function of the gene. And these extra copies of a single codon causes a faulty protein, and also expansion of trinucleotide repeat in the non-coding gene region suppress gene and suppress protein expression. Different diseases have been known by this mechanism. For example, we said that the trinucleotide repeat mutation in 5' UTR, the sequence of CGG causes Fragile X syndrome. In the exon, CAG causes Huntington disease. In the intron, GAA causes Friedreich ataxia. And in the 3' UTR, CGT causes myotonic dystrophy. What is this type of mutation?
Trinucleotide.
Yeah, trinucleotide repeats. Exactly. What is this mutation?
Frame shift.
Yes, because of the deletion or insertion of the
base.
one base per one nucleotide. This is the schematic picture for the Fragile X syndrome. In this syndrome, you can see that there is triple repeats of CGG in FMR gene. So FMR gene function have problem. It is called Fragile X syndrome because in folate deficient medium, you can see the break of the distal end of the X chromosome as a break. So this break has caused the naming this disease as Fragile X syndrome. It has a X-linked and also or male dominance. It is the females are almost carriers of this disease, and the presentation is in the males. As we see, more than 230 nucleotide repeats is required for the presentation of Fragile X syndrome. We tell you the manifestations of this disease. Results from a mutation is in FMR gene Fragile X maternal retardation 1, which maps on X chromosome, region 2, band 7, subband 3, the most common genetic cause of intellectual disability in the males, and second cause of genetic disability, mental retardation after Down syndrome over. In the second most common genetic cause of mental retardation after Down, and it predominantly affects the males. In karyotyping of the folate deficient medium, you can see this break part. Nowadays, this type of diagnosis, and this diagnosis the method has been abolished because you have very more precise methods like trinucleotide repeat PCR and fragment analysis. You can calculate the lengths and repeats of the CGGs and you can diagnose this disease very better instead of folate deficient medium and karyotyping. Culturing patient cells in folate deficient medium typically show discontinuity, staining, and construction in line arm of X chromosome. This is the mechanism disease. The repeat of this CGG cause methylation and gene suppression of FMR1. And this gene suppression has very phenotypic sequences. What are these phenotypic sequences for Fragile X syndrome? First of all, long face, large everted ears, large testicles, mitral valve prolapse, and the most important one, moderate to severe mental retardation. In the normal population, the number of repeats is 29. More than 200 to 4,000 repeats causes disease. The fold mutations happens when there is more than 200 repeats. Between 52 to 200, it is called permutation. Carriers, males and females, have permutation. You see that the carrier female can have affected male child. During ovogenesis but not spermatogenesis, permutation can convert it to full mutation. So it is very important when you look at a pedigree, if there is a mental retardation in a mother family, you should suspect these diseases. And also it can cause premature ovarian failure in the female. For example, you can take the history of premature ovarian failure in that family, mental retardation. These findings can guide you to diagnosis. This is another picture for presentation of the Fragile X syndrome. Okay, 5 minutes break.
Yes.
And disease arising from chromosomal abnormalities. First of all, Mendelian disorders resulting from mutation in a single gene. These genetic abnormalities show high penetrance, meaning that most individuals that give these mutations show the disease. The diseases are hereditary and familial. Other complex disorders or multiple genes as well as environmental factors influence these diseases. They are like hypertension, diabetes, allergic diseases, autoimmune diseases, and schizophrenia. So there is a multifactorial disease or between the environmental and genetic disorders. This is arising from chromosomal abnormality, can be numerical chromosomal abnormality or structural chromosomal abnormality. Mendelian disorders can inherit as autosomal dominant, autosomal recessive, or X-linked disorders. There are examples of autosomal dominant, recessive, and X-linked disorders. Can you give me some examples about these diseases? Autosomal dominant? We can see the names.
Oh, okay. So for example, hypercholesterolemia.
Oh yeah, hypercholesterolemia. The sickle cell, semi-dominant, recessive, it is not completely dominant. But Ehlers-Danlos, Marfan, hereditary spherocytosis, neurofibromatosis, autosomal polycystic kidney disease are autosomal dominant, autosomal recessive like cystic fibrosis, mutation in CFTR gene, phenylketonuria, mutation in phenylalanine hydroxylase, Tay-Sachs mutation is hexosaminidase, Severe combined immunodeficiency, various genes, but adenosine one of them, alpha and beta thalassemia, hemoglobin, beta chain, sickle cell anemia, hemoglobin, beta chain, X-linked like hemophilia, Duchenne Becker, muscle dystrophy, and fragile X.
If you should memorize all of them for the final exam, memorize.
Finalism, what is
You should memorize all of them.
No, it is not necessary. Just the diseases that are discussed in the next slides are important. A single gene mutation may have pleiotropy or genetic heterogeneity. What is the pleiotropy? When one single mutation has various presentations. It is called pleiotropy. For example, sickle cell anemia, just one mutation, different presentation. For example, pain, susceptibility to infection, anemia, or heart attacks, brain damage. This is caused by just one mutation. It is called pleiotropy. Whilst on the other hand, genetic heterogeneity, that means mutations in various genes can cause one presentation. For example, in the retinitis pigmentosa, an eye disease, a type of progressive blindness, more than 50 genes play a role. So it is called genetic heterogeneity. Let's talk about the patterns of inheritance and pedigree. You know, for pedigree, are you familiar with the pedigrees?
Yes.
Okay. You can draw a family tree or pedigree for at least 3 generations. For giving a genetic consultation to a family, you need to draw a pedigree or family tree at least for 3 generations, you know. And in the autosomal dominant disorders, at least one of the parents is affected. When one of the parents is affected, she or he himself show the presentation, and at least 50% of the children are affected by the disease. This is the pattern of autosomal dominant, one copy altered, half of the children are affected, one of the parents is affected. Sometimes the parent is not affected and this mutation in the child has happened de novo or by itself. You know, the parent is healthy but the child is involved. The siblings are neither affected nor increased risk of developing the disease. You know, father age is very important for de novo autosomal dominant disorder. For example, a very old father can have an affected child with autosomal dominant disorders because of the problem with spermatogenesis in old age, cigarettes, alcohol, other types of the toxic substances. So aging and environmental toxins is very important for de novo. These types of de novo mutations. Let's talk about the variable expressivity. You know that expressivity measures the extent to which a genetic variant actually expresses the so-called clinical abnormality at individual level. If a trait is consistently associated with a mutant gene but is expressed differently among persons carrying the gene, for example, neurofibromatosis 1, they have different expressivity. For example, you have neurofibromatosis but very slight capsulate spots, but the other one has neurofibromatosis, very large and huge neurofibroma in the abdomen in very critical sites of the body, severe or very mild presentation. This difference is called expressivity, which is seen in autosomal dominant disorders. This is penetrance. Yes or no presentation. This is expressivity. Difference between the severity of the expressions of the symptoms and the phenotype. This is the difference between penetrance and expressivity. A 50% reduction in the normal gene product is associated with clinical signs and symptoms. But it is not true for the autosomal recessive or enzymatic disorders. In autosomal dominant disorders can affect membrane receptors in hypercholesterolemia, transfer proteins, structural proteins such as collagen, and red cell membrane such as spectrin in spherocytosis, hereditary spherocytosis. Let's talk about one of the autosomal dominant disorders, Marfan syndrome, which is caused by a mutation in the structural structural protein. Fibrillin 1, exactly, fibrillin, excellent. This is the clinical presentations of the Marfan syndrome: arachnodactyly, very long and thin fingers, very long hand span, arm span, and also dolichocephaly and malar hypoplasia. It is autosomal dominant disorders of collagen of connective tissues cause change in the skeletal, ligament, eye, ciliary, zonus, and cardiovascular system. So mutation in fibrillary cap can have, in the Marfan syndrome, can have a skeletal manifestation, cardiovascular manifestation, and eye manifestation. It is caused by a defect in extracellular glycoprotein called fibrillin in 15 chromosome. This glycoprotein is secreted by fibroblasts and found in extracellular matrix. It serves as frame for deposition of elastic fiber, which play a role in resilience, flexibility, and elasticity of the tissue. As we said, Marfan syndrome gene, fibrillin 1, FBN1, is located on chromosome 15, Q, region 1, band 1. Region 2, band 1, more than 1,800 distinct positive mutation is found in FBN1 gene. You can see the function of the fibrillin in the extracellular matrix and the association with elastin fiber formation, which is important for resilience, elasticity of the skin. It is now evident that the loss of microfibrils give rise to abnormal and excessive activation of the transforming growth factor beta, TGF-beta. Excessive TGF-beta signaling has deleterious effects on vascular development and integrity of extracellular matrix. They are widely distributed in the body, but are particularly abundant in the skeletal, ligament, eyes, ciliary zonus, and cardiovascular aorta. One of the typical presentation of Marfan syndrome is ectopia lentis, or lens dislocation toward the upper and outer side of the eye. You know, this dislocation is seen in Marfan, the other dislocation can be seen in the other diseases. A schematic presentation of the Marfan: aortic dilation, regulation, regurgitation of the valves. It is a mnemonic, 15 on chromosome 15, fibrillin, and flat feet, arachnodactyly, Marfanoid habitus, pectus excavitus, a cavity in the pectus.
Pigeon test.
Yes, it's pigeon test, exactly, pigeon test. Excellent. And also bulla and apical blebs, pneumothorax, hyperextensible joints. They are presentation of Marfan syndrome. So in Marfan syndrome, you have skeletal abnormalities, lying legs, arms, and fingers, arachnodactyly, hyperextensibility of the joints, severe kyphoscoliosis, dolichocephaly, bilateral dislocation of the subluxation of the lens, secondary to weakness of its suspensory ligaments, causes ectopia lentis, and a cardiovascular presentation like aneurysmal dilation, aortic dissection, and mitral valve prolapse. You can have variable penetrance and expressivity in Marfan syndrome also. Abraham Lincoln was one of the most famous persons was affected by Marfan syndrome. Let's talk about the autosomal recessive disorders. Both parents, you need two affected alleles for the disease. And also, both parents are carriers of the disease but not affected by the disease. And approximately 1/4, so 25% of the children are affected by autosomal recessive disease. So if you look at the pedigree, you can guess what is the pattern of inheritance. You see that both alleles are affected in this disease. Both parents are carriers. 25% are affected, and 50% are carriers of the disease. In autosomal recessive disorders, the expression of the defect tends to be more uniform. You have complete penetrance. Onset is frequently early in life, and in many cases, enzymes are affected. So let's talk about the first autosomal recessive disorders. Have you heard about PKU? Would you please tell me what is PKU?
Deficiency in phenyl alanine hydroxylase.
Exactly. Phenylalanine is an amino acid which is essential for protein synthesis. In normal children, less than 50% of dietary intake of phenylalanine is necessary for protein synthesis. The remainder is converted to tyrosine by phenylalanine hydroxylase. So 50% of the phenylalanine taken by dietary by food is redundant and will be changed to tyrosine by phenylalanine hydroxylase. There are low phenylalanine foods like vegetables, fruits, sugars, low protein foods, special breads, and there are high phenylalanine foods, fish, meat, dairy, egg, all delicious foods are full of phenylalanine. It is difficult for patients really. Phenylalanine gene is autosomal recessive disorder located on chromosome 12. It results from mutation in phenylalanine hydroxylase. The biochemical abnormality in PKU is inability to convert phenylalanine to tyrosine. This is the metabolic pathway of the phenylalanine. Phenylalanine under effect of phenylalanine hydroxylase is changed to tyrosine with cofactor of tetrahydroxyopetran. Tyrosine under the effect of tyrosine hydroxylase is changed to dopa. Dopa under effect of tyrosinase is changed to melanin, or dopa decarboxylase to dopa. Catabolism of tyrosine. At the molecular level, approximately 1,000 mutations have been discovered in this gene. Only some of which cause a severe deficiency. Only some infants with some residual activity present with milder type of disease. It is called benign hyperphenylalaninemia. For the severe type of disease, the level of phenylalanine should be more than 5 times than the normal. Phenylalanine or its metabolites excreted in large amounts in the urine and sweat. And the baby affected by the PK, you have a special odor like a mouse, yes, mouse or mouse eater. Brain damage happens by the age of 6 months. It causes severe mental retardation. Untreated phenylketonuria children cannot walk in 1/3 of cases, and 2/3 cannot talk if they are not treated by special regimens or drugs. It can also cause seizure. Concomitant lack of tyrosine, precursor of melanin, is responsible for lighter chain of hair and skin and eczema. So they have very fair complex, because of light color of hair and skin. This is schematic presentation of the PKU, seizure, eczema, intellectual disability, hypopigmentation, light hair color, mouse odor, autosomal recessive. Serum phenylalanine level is used to differentiate benign phenylalaninemia from PKU. The level in the PKU typically are more than 5 times higher than the normal. After the biochemical diagnosis is established, the specific mutation causing PKU can be determined. Enzyme replacement therapy by phenylalanine ammonia lias that convert phenylalanine to ammonia, a non-toxic metabolite, can be used as a treatment, and also reduce circulating phenylalanine in classic PKU. And dietary restriction is also very important for treatment of the PKU. 98% of cases of hyperphenylalaninemia are attributed to mutations in PAH. The rest of 2% are because of abnormalities in recycling of the cofactor tetrahydrobiopterin, and also it is very important because the enzyme replacement and dietary restriction is not effective in these cases, and this cofactor can be given as a drug, and also treatment is very effective. So it is important for you to diagnose the mechanism very exactly. Clinical recognition of these variants forms is important because the patients cannot be treated by dietary restriction of phenylalanine, but also requires supplementation of tetrahydrobiopterin or BH4. Let's talk about the X-linked disorders. Not very important disease-causing genes are located on Y chromosome except the sex-determining genes like AZF, the deletion of a mutation of AZH, can cause azoospermia, infertility in men. Except these genes, no very important genes. Most of X-linked disorders, sex-linked disorders are located on the X gene, X chromosome. X-linked disorders are characterized that a heterozygote female can have affected male sons, and also affected male can have carrier daughters. Okay, we talked about the Marfan syndrome. This is caused by mutation in genes encoding the structural proteins of Marfan syndrome. This is caused by mutation in genes encoding receptor proteins and channels are familial hypercholesterolemia or cystic fibrosis. This is caused by mutation in gene encoding enzyme protein or PKU, galactosomal lysosomal storage disease, like Tay-Sachs, Niemann-Pick-Gaucher, and also mucopolysaccharidosis and glycogen storage disease. Let's talk about the Ehlers-Danlos syndrome, another autosomal dominant disorder, the disorders that cause defects in collagen synthesis can affect various organs like the skeletal ligament, and also the examples are Ehlers-Danlos, osteogenesis imperfecta, and epidermolysis bullosa. They can have autosomal dominant or excessive recessive patterns. Approximately 3 distinct types of collagens are present that products of different genes. Different collagen genes are present. Mutation to some extent, the clinical heterogeneity of Ehlers-Danlos is explained by mutation in different genes that produce the collagen. We have at least 6 types of the Ehlers-Danlos syndrome, and in this disease, the tissues that are rich in collagen are affected, like skin, ligament, and joints. They have very flexible and hypermotile joints in Ehlers-Danlos syndrome. Skin is extraordinarily stretchable, extremely fragile, and vulnerable to trauma. The structural failure of organ and tissue may lead to serious internal complications. For example, rupture of very large vessels and colon is one of the complications of Ehlers-Danlos syndrome, or rupture of corneal and retinal detachment. We said that we have 6 types of Ehlers-Danlos. One of them is type 3 deficient synthesis of collagen. Type 3 that causes the vascular Ehlers-Danlos syndrome. Can you name me the first 4 types of the collagen and the tissue that they are present in? Collagen type 1?
Type 4.
Type 3.
Skin. Type 2, cartilage. Type 3, vessel. Type 4, basement membrane. As a mnemonic, remember please, scab, crust on the ulcer. The scab, skin, cartilage, artery, yes, basement membrane. We are talking about the collagen type 3 in the vessels. You know, this type of Ehlers-Danlos is vascular Ehlers-Danlos that causes weakness of tissue rich in collagen type 3 and predisposing to rupture of the vessels. Deficiency of the enzyme lysyl hydroxylase, which causes kyphoscoliotic Ehlers-Danlos syndrome because it is an autosomal recessive disorder and causes congenital scoliosis and ocular fragility. Most contortionists have some form of Ehlers-Danlos disease. So, let's talk about disease caused mutation in gene encoding receptor proteins, familial hypercholesterolemia. Do you have a family with high cholesterol level? No one?
No.
Patients with familial hypercholesterolemia develop excessive levels of serum cholesterol. Very excessive level of cholesterol can cause xanthoma, yellowish plaque around, for example, the tendon insertion and also the eyelids, and premature atherosclerosis in the vessels. 2 mechanisms for the production of cholesterol. One of them is endogenous. In the endogenous mechanism, VLDL is produced by the liver under the effect of lipoprotein liposphate, a free fatty acid. Is separated and used as energy source in the peripheral tissue. The remainder is called IDL. IDL is changed in the LDL. LDL is a source of cholesterol in the body. Another pathway for production of the cholesterol is the exogenous. After taking the lipid-rich diet, for example, fast foods, you have very large amount of chylomicron in the body, and the circulation under the effect of lipoprotein liposphate free fatty acid is used as an energy source. The remainder is chylomicron receptor. Under the effect of LDL receptor, it will be deposited in the liver. So there are different mechanisms, endogenous and exogenous. We explained this mechanism. LDL receptors are widely distributed in the fibroblasts, lymphocytes, smooth muscle, and hepatocytes. So the liver plays an extremely important role in LDL metabolism. Role of liver in the LDL metabolism by LDL receptor, it is absorbed in the endosome, fuses with the lysosome, and the free cholesterol under the effect of enzyme is produced. When the free cholesterol is increased, it can be stored in the liver or suppress HMG reductase, which produces the cholesterol, or it has a negative feedback effect on LDL receptor. HMG reductase is the target of drugs, statins. For example, atorvastatin, rosuvastatin, the drugs that used for reducing the cholesterol level in the blood. The other mechanism for uptake of cholesterol is LDL is a scavenger receptors on the mononuclear macrophages and phagocytic system that activate and oxidate the LDL. Patients with familial hypercholesterolemia develop excessive levels of serum cholesterol, and they have premature atherosclerosis in the autosomal recessive form. It can cause myocardial infarction and death before the age of 20 if it is not diagnosed. Okay, let's talk about the cystic fibrosis. 1 minute inside to rest.